I'm assuming that if you did the mini exam last time, you're going to do it again. You're staying with that. If you want to switch to the sectional exams, you need to let me know. And if you did the sectional exams, you want to switch to the mini exams, you need to let me know in case today. Okay. All right. So we're going to finish sensory systems today, but first we got to finish vision before we can get into hearing and the chemical senses. So we left off here talking about receptive fields, right? Big takeaway point of receptive fields are that we have way more receptors than we have neurons that can go to the brain. 126 million to 1 million, right? So we have to have convergence. We have to take a whole bunch of receptors and get them down into one output. And so by having a baseline signal, right, so that when nothing is happening, the neurons just kind of popping off some action potentials every so often, that allows us to send two signals to the brain, an excitatory signal and an inhibitory signal. We can go down for baseline. Okay? So here we have baseline, nothing happening, right? Excitatory signal, inhibitory signal. Two signals, and that allows us to know whether the light is in the center or the surround of the donut-shaped receptive field. Okay? We got to take all these cells and converge them down into one. By having two signals, we at least get two pieces of information. We know if it's in the surround or the center. Right? And it doesn't matter if the center is excitatory or if the center is inhibitory, the surround is always opposite. Good. All right. So, I want to talk a little bit about neural coding in the visual system, which is complicated. So we're going to focus on color and what is the neural code for different colors as an example of how neural coding works in vision systems. All right. So originally, people were able, by doing experiments with humans and saying, hey, if I take these two types of light and I mix them together, what color do you see? And it turns out that by doing this, they could take three different lamps, colors of light. We could generate every possible color that humans could see. And that's because we have three types of photopigment in our cones, short, medium, and long wavelength, and that's called trichromatic color coding. So if my short wavelength cones are sending a signal, that's labeled line for blue, right? And if my medium wavelength cones are sending a signal, that's labeled line for green. And if my long wavelength cones are sending a signal, that's labeled line for red. Do we see more colors than blue, green, and red? Yes. So like most sensory systems, we have this labeled line for specific colors, but by looking at the pattern, right, if I have activity, 8 action potentials coming from my medium and 8 action potentials coming from my long, that's saying green and red. What color is it? Y'all don't know this right here. What color is in between green and red? What color would equally stimulate green and red?
Yellow.
Yellow, thank you. Yellow, right? So I can look at the pattern of the trichromatics and say, okay, that's yellow. What if all three are equally stimulated? I have 8 action potentials from short, 8 from medium, 8 from long. All of them are equally stimulated.
Black. No, white.
White, right? If the 8 went down to 2, then it would be dark gray, right? If there's nothing, it'd be black, okay? You see how this works, okay? Now, it turns out that when we get to the LGN, the thalamus, when we get to the level of the thalamus, the trichromatic, these 3 outputs, have actually changed through hard wiring and have become what we call a phonic process. So now we have neurons that are stimulated by red but inhibited by green. were stimulated by green and inhibited by red. They're paired up in opponents of complementary colors. And so we have red versus green, and we have blue versus yellow. So we go from 3 outputs to 4. Now we have a yellow. Right? And so we have, if blue is active, then yellow is inhibited. Yellow is active, blue is inhibited, and red versus green. Okay. So as I mentioned, the receptor output, by looking at that, the size of the cones is sort of indicating how many action potentials are coming from each of these. If we have blue, right, then only short wavelength are sending a signal. If we have red, only long wavelength are. If they're equal, we have white, right? If we have equal amounts of medium and long wavelengths, we perceive that as yellow. Okay. Good? Good. All right. So this is just showing how that would work. If I have 3 slide projectors and I have red, green, and blue light, right? Red and green, we combine those, and what color do we get?
Yellow.
Thank you, Gracie. So we have equal stimulation of medium and long wavelength light here, right? Yellow. All 3 of them, we get white, right? Okay. So at the first stage, at the cones, we have short, medium, and long wavelength. But then they send excitatory and inhibitory projections to the thalamus, to the LGM of the thalamus, that generates 4 different signals. And these signals are oppositional: red, green, yellow, blue. I'm going to process them. Good? All right. So if we have equal amounts of blue and equal amounts of yellow, what's going to happen? We have 5 excitatory blue and 5 inhibitory yellow. That becomes what? That's division. We have 5 excitatory, we have 5 here and 5 there, right? The blue is excitatory 5, the yellow is inhibitory 5. What happens?
We say cancel.
They cancel each other out, right? So if I take a square and I say be yellow at the top and be blue at the middle and mix it in between, what happens when we get to the middle where it's equal amounts? It becomes grayish or white, right? We lose, they cancel each other out, right? Cancel each other out. And so it's one of the ways we know about opponent process. All right. All right, are we good? That's neural coding. Trichromatic becomes opponent process. The brain has to look at the pattern in order to know. Obviously, it's easy to know blue, yellow, green, and red because we have labeled lines for those, but anything that's not exactly those colors, we have to look at the pattern to know where it falls, right? Orange is going to be some yellow and some red because orange is in between yellow and red, right? Teal is going to be some green and some blue because it's halfway in between those. Yes, right.
The opponent process was labeled line and trichromatic was which one?
They're both labeled line, right? But the brain has to look across the pattern of labeled line inputs in order to discern colors that aren't blue, yellow, green, or red. Okay. All right. Pathway. We have 2 visual fields. We have a left visual field and we have a right visual field. Now, it gets tricky because when we have there's no point. Okay. The optic nerve from each eye, right, is going to project to the thalamus because all sensory systems run through the thalamus. The part of the thalamus that vision runs through is the LGN, lateral genicular nucleus, LGN. And it also sends input to 2 other places that's beyond, that's not at a level of our perception, right? So from the thalamus, we're going to go to the occipital lobe for perception, vision perception, right? But we also send input to the superior colliculus. What does the superior colliculus do?
Pattern vision.
Head orientation to visual stimuli, eye tracking, right? Stuff we're not like consciously aware of. And signal gets sent to the hypothalamus. Anyone remember what the hypothalamus is involved with? Types of processes. We're going to talk about them starting on Thursday. Third, our internal states. Why would we send vision, a signal from our eyes? What internal state might care about that?
Consciousness?
No, but yeah, kind of. But no.
You like to see food or you like to see what we want?
No, you guys are good guessers. It's our sleep-wake cycle, right? We want to know if it's daytime or nighttime, right? So it helps regulate our sleep-wake cycle that's controlled by the hypothalamus. All right. So this is where I was trying to go. If we have a point in our right visual field, okay, that point is going to actually project to both eyes, right? If I put my hand out here, right, and I close my right eye, I can see it. I close my left eye, I can see it, right? It's going to both eyes, okay? So we have two images of that thing. We have to make them meet up. That's where the optic chiasm comes in, okay? Why do you have to learn optic chiasm? Because it's important because the nasal, the medial part, the nose part of each eye crosses over, and meets up with its partner from the lateral part of the other eye that stays on the ipsilateral side, right? So this guy stays ipsilateral, this one crosses over, and they meet up so that I have one representation of this thing in my brain. So everything in my right visual field, even though it's going to both eyes, ends up in my left occipital lobe. And everything in my left visual field is going to meet up and go to my right visual cortex. Okay. So that's the way we consolidate. We consolidate the visual field going into both eyes by crossing over the nasal part of each eye, crosses over and meet up with its partner from the other eye. So that we only get one image. Good? Okay. All right. So that's the way that works. All right. So we end up having contralateral perception, right? Left visual world, right brain, right visual world, left brain. This is just another picture of how that goes, right? Primary pathway is going to V1, visual 1, in the occipital lobe of the cortex of the brain. There's your part of the brain. We do have those other secondary pathways for eye tracking and spirit folliculus and for sleep-wake cycle regulation in the hypothalamus. All right. How is the brain organized? Just like we talked about touch being organized by dermatome, right? In areas, we have the same sort of thing, except this is organized by our retina. It's what we call a retinotopic map. So if something's over here, right, that's projecting to a part of our retina, and we're organizing that by columns in this V1 area. Now, V1 is going to break down these complex shapes into primary features, and then we sort of rebuild our perception in the secondary areas of the brain. So in V1, we're detecting primary features like angles of lines. So for a particular place, receptive field, I might have columns that respond to horizontal, all the way to vertical, all the way back to horizontal, and any angle of line in that particular place. It's going to cause that column of neurons to fire with activity. And so we reassemble all these elements of what we're seeing back into this complex picture, right? So we have things like line orientation, angles of lines, simple movement, patterns, all being represented. So, as I mentioned, we have these secondary areas that are now going to take those primary features, look at that pattern of firing in V1, and assemble it back into objects. So we have areas called V2 and V3 that are going to do object and form perception. We have a special area V4 for color processing, for identifying colors. We have special areas V5 and middle superior temporal cortex that are doing movement perception. So we have dedicated areas of this. These are secondary vision areas that are dedicated for particular complex processing, right? And in general, we end up with two pathways out of V1. We have a ventral stream that's going to go down into the temporal cortex. What is the temporal cortex involved? What do we find in the temporal lobe? Memory. So that ventral pathway is identify what this object is, associate the associate this object with an eraser, right? You know what an eraser is? It's a memory. So when you see this, the ventral pathway is identifying the shape and finding the memory that belongs with that shape. Okay? The dorsal pathway is coming up through the parietal lobe to the frontal lobe. What's the frontal lobe do? A lot of stuff, right? What's it do? Yes, decision making, right? And what else is a big part of the frontal lobe that we talked about last week? Motor movement. So this pathway is what do I do? How do I respond? What decision do I make? What motor output do I do in response to that visual stimulus? So we have a what do I do with it and what is it pathway. An identification and an action pathway. All right. So dorsal stream is, sometimes it's called aware stream. I like to imply it's called aware stream. But think of it as an action stream. Think of it as going to the frontal lobe to do something with that visual stimulus. How do I respond? And then we have a what is it, an identification pathway into the temporal lobe. We have some specialized areas like this fusiform gyrus down here that's dedicated to emotionally important visual objects. And what are some of the most emotionally important visual objects in humans? Faces and people, right? So this is a place that's sort of dedicated to facial recognition and identification of people's faces. This fusiform. All right. Yes.
Why does that say parietal lobe? Because it goes into the frontal lobe?
Yes, because it goes through the parietal lobe here, right? Towards the frontal lobe. The parietal lobe is actually still like some vision processing before it gets to the touch area. All right, question of the day. We have time to speak. We're going to make time. We're going to try this. It's very interesting. So, when that area, the fusiform area, gets damaged, it's tucked away deep down here. It's hard to damage that, right? It's pretty protected. It's underneath the brain, right? It's not sticking out here against the skull. So it's pretty hard to get damaged. So it's pretty rare that gets damaged. But when it gets damaged, people have problems recognizing faces. And that's a condition called proboscidnosia, big old word. And we're going to meet Terry Sweeney, who has proboscidnosia.
Meet Terry Sweeney, a professional photographer. She was a firefighter until an injury damaged part of her social brain, the circuitry she uses to recognize faces. The disorder is called prosopagnosia. For example, the person Terry has been photographing here is her mother. Now, because of her injury, Terry can't recognize any face.
Please not.
Not even her own mother's. In this test, Terry takes a Polaroid picture of her mother.
Terry, you just took a picture of your mother.
If you take a look at these and see if any of them are the picture you took or not, maybe.
Now, Massachusetts General Hospital researcher Dr. Nancy Eckhoff shows Terry several Polaroids with only faces showing and asks Terry to pick out her mother.
Okay, if you had to guess, which looks to you most like your mother? Just looking now, you can't tell which one of these now is really like your mother to you.
No.
Okay.
If you just go off the face, I just look at the clothes and I can tell you.
Okay, do you want me to uncover them? Okay, why don't I do that? I'll uncover them so you can see what they're wearing up across the picture.
See, that's where it's good. The pink and purple shirt.
The shirt. You're looking at the shirt.
It's the hardest, I think, with my family, not to be able to personalize them with a face or a sense of timing. You know, it's kind of like I've lost everybody, like everyone's gone, and all that I'm really left with are facts without faces. You know.
When Terry looks at these slides of famous faces, she can't recognize them, although other cues like hairstyle prompt a guess.
Madonna. Because her hair, look at her hair.
It's Marilyn Monroe.
Really? I always feel like I'm struggling and always feel like I'm fumbling through myself to find a connection, to find a bridge that'll just pull everybody right back all of a sudden.
Can you tell who that is?
No.
Okay, they said Bruce Springsteen.
That's not Bruce Springsteen, is it?
Why do you say that?
This guy's ugly.
Now take a look at that woman right there. Does she look at all familiar to you?
No.
What if I were to say that's you?
I better not be me.
It is.
Really?
All right, so when this specific area gets damaged, you lose this ability to assemble. She can see eyes and nose and face, but can't put it all together and can't associate it with what it belongs to. Now, if her mom speaks, she recognizes her voice and immediately knows who it is, and she can then learn, okay, my mom's wearing a purple shirt today, I see the lady with the purple shirt today, that's my mom, right? And so there's cues that she can use to pick up on to help identify things. All right, good on vision. All right, so we're going to go through the auditory system and through smell and through taste. And again, I wrote this up here during the video clip. These are the things that we're focusing on, right? These are the things that we're focusing on. So what's the physical stimulus for hearing? What do we detect? Sound waves. Sound waves. Changes in air pressure. And we, just like we talked about with light, right? There's a lot of electromagnetic radiation. We see a narrow slice of that from blue to red, okay? With hearing, we don't hear all frequencies of sound waves. Our sound waves are, we hear from 20 hertz, meaning 20 compressions per second, up to about 20,000 hertz when your hearing is perfect. Two important parts of sound waves, right? The wavelength, right? How many cycles, how many compressions there are is, in hertz, is related to whether it's a low sound or a really high sound. Right? But the higher the frequency, the higher the pitch. So the tone of the sound is related to the hertz, is governed by that. The size then, how much compression there is the volume, is how loud is that sound, the amplitude. So here we have the same size, amplitude, but a low frequency, that would be a lower tone, and a higher pitch tone, but they're the same amplitude, right? We can also have the same wavelength, low amplitude, that would be a soft tone. This would be the same tone, but louder. Good. All right. So, check off physical stimulus. Receptors and transduction. There's actually a multi-step process to get these sound waves into our cochlea, which is where transduction is going to happen. So the first thing is we have these funny looking things called pinna, and these pinna are collecting sound. They're helping capture the sound waves and funnel it down into our middle ear. Okay? The reason that our earlobes are shaped in this particular way is the shape actually helps us localize vertically where sounds come from. I'm not going to get into how we do that. We talk about that in sensation perception. We'll have to stay tuned for that.
I was just gonna ask, some people's ears stick out farther from their heads. Does that impact it in any way?
Yeah, right? So we have different physiologies, right? And so, and you're born with that, and so you sort of learn, I mean, that we talked about, we didn't really talk about that, but yes. All right, we get the ear captured by the pinna, funneled into our middle ear. Our middle ear has one job to do, and that is to amplify the airwaves and project them onto the cochlea that's full of fluid, right? So if I flap my arms like this, I'm making compressions of air. You can't hear it because I can't move my arms faster than 20 times a second, right? But I'm moving air, I'm compressing air, right? Now, if I'm standing in a pool up to my neck and I'm doing the same thing, is it easier or harder to make the same compressions in water? A lot harder, right? So we're taking air compressions and we're using the tympanic membrane, or your eardrum vibrates with that movement of the air. And then we have these three bones, the hammer of the animal and the stirrup or stapes, and those three bones act like a lever to increase the force of that air movement and replicate it on a fluid-filled cochlea. If we just had that air coming in directly onto the, trying to stimulate, trying to move the fluid in here, it wouldn't have enough force to do that. We have to amplify it with those bones. They do this lever-like action. Okay? Anyone plays trivia in here? Those are the three smallest bones in the human body. It's the bones in the middle here. Okay, and then we finally we've replicated the compressions in the fluid-filled cochlea. It looks like a snail shape here. And so now that's where transduction is going to happen. So that vibration now is transmitted through the fluid of the cochlea. Here the cochlea has been stretched out so we can kind of just see how it works. It's kind of hard to see it when it's all like a snail shell. But it vibrates down and around, and depending on the hertz, depending on the frequency, it's going to stimulate a particular place on the cochlea. And so it's the innermost part are low frequencies, and it goes all the way up to 20,000 hertz by the outermost part. So where on the cochlea gets stimulated corresponds to the tone that's present. These are just some other pictures of how this happens. And what happens is, as that fluid wave that's vibrating at a particular frequency, moves through these ducts, it causes this thing called the tectoral membrane to bounce up and down on hair cells. And if you remember back to the vestibular system, when we talked about force opening and closing those channels, it happens the same way. It's the force of the tectoral membrane bouncing up and down is going to allow that fluid to come in. And again, here it's potassium, but potassium is positive. That's going to depolarize those cells and we're going to release neurotransmitters. So they're set up with those little amino acid chain link chains there, so that if it bends in one direction, it opens the channel and potassium comes in. And if it bends in the other direction, it closes the channel and doesn't, and we get no signal. And so it's the pushing down, the stimulation of that textual membrane bouncing on those hair cells that causes them to fire. Yes?
Does everybody hear the same, like with your voice, like does everybody hear the same tone of your voice?
Yes. So I'm going to say that there are individual differences, and generally yes. Yeah.
Okay, when you're in an airplane and you feel your ears pop, what's happening there? Because I've heard people that start bleeding,
That's-
pressure doesn't get relieved.
Right, that's because, right, your tympanic membrane, right, you have pressure on either side of that, and you have to equalize that pressure. Otherwise, you can rupture your drum and things can, because there's air on either side of that. Right? That's what you're talking about.
So when the membrane is pushing on the hair cells move one way or the other.
Yes.
Okay, so there's two ways they can move, and
Yeah,
one
so
way
it pushes down.
is open.
Let's say we push it down, it moves it that way, it lifts up, and it rebounds and goes the other way. And if nothing's happening, it's just sitting there like this. Yes.
So what is the receptor?
The receptor are these hair cilia cells, and the transduction, the taking the physical force of the sound wave, it's now a fluid wave, and turning that into a biological signal is potassium comes in, depolarizes the cell, and we get neurotransmitter release. Good? Good. Okay. So as I mentioned, where on the cochlea is stimulated corresponds to a frequency, corresponds to a tone. And the innermost part is 20 hertz, and it goes up all the way to 20,000 hertz at the outermost part. Okay? What do you notice about the pattern of the stimuli there? You got low frequencies here, and we're going all the way to 20,000 hertz here. Yeah, so we were like at 500, 1,000, 2,000, 5,000, 7,000, 20,000.
So two things, when there's one in the middle of the gaps are bigger, so the hertz is lower, or like the hertz is lower, and then at the end the gaps are smaller, so the hertz are higher.
Yeah, so I think what you're trying to say is that it's unequally distributed, right? Just like the homunculus, right? We give more area to certain frequencies than others. So look how many receptors we have for things all the way to 5,000 hertz. All the cochlea over here is 20 to 5,000. And then from here to here is 15,000 hertz, from 5,000 all the way to 20,000. So we give most of our cochlea is dedicated to sounds under 5,000 hertz. Why would we do that? I don't know.
Like certain noises can damage.
Sure, and so the high frequencies are most susceptible to damage because they're sticking out here near the periphery, and these are less likely. Aaron, do you have a guess?
Well, I don't know, I guess I was thinking we associate loud noises with threats or something that's not supposed to be happening, and that usually around a lot of loud noise, so we want something that gets our attention.
Yeah, but we're not talking about amplitude, we're talking about hertz here. Why would we care about sounds less than 5,000?
It's like that's where most of the sounds go lower. It's separate from
Not most of the sounds, but the most important sounds.
human voices.
Yes, human language, right, is primarily under 5,000 hertz. And most of the frequencies associated with language are under 5,000 hertz, and so we devote, most of our hearing is devoted to understanding language, okay? Yes.
So what would be something that's 20,000 hertz?
Very, very high percent.
Okay, so what would be like a 5 kiloton?
A lower tone because you can hear. It's loud, but it's not, but it's a range we can hear. 20,000 hertz would be very difficult to hear.
So that'd be something that would also hear what we
Yes. Okay. The more receptors we have, so here's our sensitivity, right? We have the most receptors in this range, therefore we have the best detection, the lowest threshold for them, right? Other species, as we just mentioned, right, have different ranges of hearing. So a dog whistle that you blow that a dog can hear produces a frequency above 20,000 hertz. So we can't detect it, but it's still within the range that a dog can detect it. Yes, Ken?
When you get like older and you can't hear those high pitches.
Because you've damaged that part of the couplet that's closer to the outside. Yes. All right, neural coding. The first idea of neural coding was, hey, if we're bouncing on these hair cells and producing action potentials, the action potentials could line up in frequency, right? The problem with that is that once you get above 100 hertz, right, we have that refractory period, and we can't fire action potentials 5,000 times a second, right? So that idea didn't really pan out, but what we realized is that if we take a whole bunch of neurons that are all responding to that same frequency, and they're all sort of randomly firing at peaks, and we look at all of their activity, we send a volley from this group of action potentials that approximates the frequency. So no one neuron can fire that many times, but if we have a couple hundred neurons that are all firing at the peak, the activity of that group of neurons can match up and represent that hertz. So we can do that up to about 5,000 hertz, and after 5,000 hertz, it's too fast to do that. Okay? So we have two types of neural coding. The place on the cochlea, the group of neurons that are being stimulated by that place, is label line for a particular frequency, right? Because it's organized by frequency around the cochlea. So there's a place for 100 hertz. Plus, so that happens for everything. Above 5,000, there's a place for 7,000. There's a place for 10,000. So any activity in the neurons at that place represents that frequency. Okay? That's label line. But we also, under 5,000 hertz, can look at a volley from a group of those neurons and sort of count the number of action potentials to know the frequency. And that gives us better discrimination between tones that are close to each other. They're close in place, but because that we can look at the volley, we can actually count and see 25,000 versus 25,005 hertz. And we can hear a difference of 5 hertz there, even though the places are right next to each other, because the brain's counting the volley coming in, the action potentials coming in, we can discriminate and hear 2 different sounds. Cool. Place, volley. All right, pathway. Good on neural coding. Great. So, cranial nerve 8, vestibular cochlear nerve, comes in, the auditory signal comes in, ends up at the cochlear nucleus in the brain stem. Right? From the cochlear nucleus, I have an abbreviation to help you, a mnemonic to help you know where it goes. Sonic MG, right? It's going to go first to this area called the superior olivary nucleus, the SON. The SON matches up the time that sound hits our ears and does this amazing calculation, and we can tell down to about 10 microseconds difference if a sound hit my right or left ear first, and that helps us localize sound. If the sound's over here, it's going to hit my right ear before it hits my left ear. So the timing of when sound hits our ears is done by the superior auditory nucleus. Then it goes to the inferior colliculus. What is the inferior colliculus involved with? Yes, orientating our head to sounds. Superior colliculus, vision. Inferior colliculus, hearing. And then it goes to the thalamus. And now we're going to the medial geniculate nucleus. So vision goes to the LGN, lateral geniculate nucleus. Hearing goes to the MGN, medial geniculate nucleus. And from there, it goes to the auditory cortex, which is in that superior temporal gyrus, the uppermost temporal gyrus.
So the medial geniculate nucleus is in the thalamus?
Yes,
That's the part of the thalamus.
the part of the thalamus that it goes through. So right now, if I ask you, can you hear the air conditioning? You can hear the air conditioning. But a second ago, your thalamus was filtering that out, and you weren't paying attention. That wasn't interfering with hearing my voice. You were saying the air conditioning is not an important stimulus. I'm going to filter that out and suppress that so I can hear his voice better. Job of the thalamus. All right. And in general, the signal's crossing over, and we're getting our right ear goes to our left superior temporal cortex, and our left ear goes to our right contralateral. All right. Just like your vision is organized in columns, and it breaks it down into features of vision like angles of lines and movement, same thing with sound. And the primary feature of sound is the tone that's present, the pattern of tones that are present. And so we have the areas of primary auditory cortex, the superior temporal gyrus, are organized by frequency. There's areas for low frequency, and as it goes on up, those areas spike as they're stimulated from the code. All right. Secondary processing. All right, primary area. Superior temporal gyrus, its function. Stimulated by frequency. All right? Secondary. We have when the brain recognizes the pattern of those frequencies it's firing as language, it sends those sounds, it sends that pattern to an area right back here called Wernicke's area that is for language comprehension. It's for language comprehension. There's also areas of association cortex, like the angular gyrus, that's specifically responsible for speech recognition. But we're also stimulating circuits down here to identify what those words are. So if I say apple, right, what happens in your head? You see a picture of an apple when I say apple? You know what an apple is? You're grabbing those memories, how it tastes, what it looks like, is it red, is it green? Yes?
So you're saying like when the sound's coming in, its first job is to do the frequency, like that's the primary job that it has, and then it goes and does the auditory cortex?
Yes, so the brain says, hey, that frequency is somebody talking. I need to, in order to understand that, I'm sending it to this specialized area that does speech recognition called Wernicke's area. And then once Wernicke's area recognizes the frequencies apple that make up the word apple, right, then it's going into the temporal lobe and grabbing what does apple mean? What does it mean? Right? All right. So because Wernicke's area speech recognition is so complex, we devote a single part of the brain to do that. It's what's called lateralized function. We'll talk more about lateralization in the next month or so. But the left hemisphere is lateralized, so all that speech recognition is happening in 90% of people's left hemisphere. Not in everybody, but in 90% of the people that we study, we see language is found in the left hemisphere. Okay? The right hemisphere, sometimes we say the right hemisphere does music. It really does pretty much everything else. Environmental sounds, music, anything that's not language. Sounds that aren't language. The right hemisphere is identifying those and working on it. So speech is so difficult, we lateralize it to the left, everything else kind of gets to the right. Yes?
Sorry, I just want to ask, did this contribute to like one side of your brain is artistic and one side of your brain is logical? Is that what
Yeah, that really isn't the case. That's like a myth, but yeah, this was sort of part of what started that, right? It's the fact that language is in the left and other stuff like music is in the left. That is true. Yes, sir?
With language and music, there's language and music.
Yeah, so when there's language and music, we get left and right brain both being activated at the same time. Right? Great. All right. An example, I want to give you an example of how we do some complex processing. I want to talk about sound localization as that example. Okay? So we localize sound. We localize sound 3 different ways. What's a superior elevator nucleus do?
Like the timing.
It detects the time that sound hits each ear. Right? So that works for any frequency sound. Right? If someone stands right to my 90 degrees to my right, that sound is going to hit this ear about 600 milliseconds before it hits this ear. That's the biggest difference we can have. If someone's directly in front of me, it's going to hit both ears exactly at the same time. And that's going to help me know someone's right in front of me or someone's right over here. And that works no matter what the frequency of sound. Time difference, all frequencies. Superior elevator nucleus, yes, Anna.
So that's asking the question of like, is it more on my right ear or my left ear? Like if I need to like localize it, if it hits my right ear first, like I know that.
Yep. Your superior elevator nucleus says it's over there.
That's what's letting me look at you right now?
Pardon?
That's what's letting me like turn my head to look at you while
Yes,
talking?
yes. Right? And yes. So we then have 2 other ways that we help localize sound. One is best for high frequencies, and one is best for low frequencies. So time works for all frequencies. Okay? But intensity difference works best for higher frequencies. So when I have a high frequency sound, there's lots of compressions of air. As it hits this ear first and travels over to this ear, some of those compressions get absorbed by my head, and that sound is actually a little bit softer, right? We lose some of the amplitude in my second ear. So we can also tell a difference in loudness between the 2 ears, and that works really well for high frequency sounds because there's lots of compressions, and the more compressions you have, the more interference our head can do. Okay? We call that an acoustic shadow, right? And the biggest difference is going to be when sounds are over here because it's more, my head's going to absorb it. And when sounds right in front of me, there's going to be no difference because it's hitting at the same time, there's no interference. With low frequency sounds, we can actually detect what phase that wave is in. Is it in the upswing, the downswing, the peak, the valley? And we can tell that by how it pulls our eardrums. It's pretty complicated. But just know, right, that a low frequency sound can be in a different phase, going up, coming down, right? And so for low frequency sounds, the intensity won't be any different, right? Because low frequency sounds don't get absorbed very well. That's why when you're in a car at the light and you hear someone's stereo, you hear the bass, you hear the thump, 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 you can't really hear the treble and the higher pitches because they're getting absorbed by other cars and other things, right? But low frequency sound travels through things and doesn't change its intensity, but we can tell the phase difference. So time difference, everybody. Okay? High frequencies, intensity difference, low frequencies, phase difference. Good? All right, there's your review points for sound. Let's get our feet. Come on, we can do it. We're going to fit it all in today. Let's go.
Are you ready for your jam in a minute? Yeah! All right, stand up. First thing, we're going to pretend like we got a jump rope.
All ready, jump rope.
Ready, go. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. All right, hands out, feet apart. Pretend like you got a hula hoop. 10 times. Ready, go. 1, 2, 3, 4.
I'm not sure about this exercise, but I guess it's expected.
7, 8, 9, 10. All right, feet together, hands on your side. We're jumping left and right. 10 times. Ready, go. 1, 2, 3,
Then some rows.
4, 5, 6, 7, 8, 9, 10. Feet together. We're jumping the feet apart and jump back in. 10 times. Ready, go. 1, 2, 3, 4, 5, 6.
All right, we're getting some glucose going. It's going to be awake for the last 30 minutes.
Reach down and touch your toes. 10 seconds. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Great job, everybody. Give yourself a hand.
All right, what's our healthy tip?
If you don't eat or drink milk products, you can get calcium from calcium-fortified cereal, breads, or soy beverages.
All right, for you lactose intolerant people.
That wasn't a two-word one.
That wasn't a two-word one.
That was a two-sentence.
It was. All right. We're talking about the chemical senses, which are two sensory systems, smell and taste. Both of them, they're grouped together as chemical senses because their physical stimulus are chemicals in the environment, chemicals in the air, go into our nose, chemicals in our mouth, go into our taste buds. Okay? Two separate systems, don't think it's one thing. Two sensory systems, but they're kind of grouped together. All right. They're the most primitive of the sensory systems, right? What is the job of smell and taste? Yeah, we want to spit out stuff that doesn't taste good, that's either rotten or poisonous, right? Most poisons are bitter. Do you like bitter things? Probably not. You probably can learn to like black coffee or learn to like bitter things, but you innately don't, okay? And what about our sense of smell? Yeah, it helps us. Are we in a good environment or bad environment? You walk into your roommate's cubicle and they haven't done laundry in 2 weeks. You're like, this is not a good environment. I don't want to be in this environment. I want to leave this environment because of the smell. Right? There's something rotten in here. Okay. So, I'm sure none of your roommates will. So olfactory stimuli are chemicals in the air. There are tens of thousands of different olfactory stimuli, and we actually have about 1,000 different types of receptors in our nose to recognize things. Taste or gustatory stimuli, chemicals that end up dissolved in our saliva. So your mom was right. If you don't chew your food, you can't taste it. If you just bypass that whole tongue sensory system, you're not tasting your food. If you chew your food and get those chemicals into saliva, you can really taste it better. Right? And we have some primary tastes that we identify. Sweet, sour, salty, bitter. Anyone ever heard of umami? Say umami. Umami, right? So that's, you just learned a Japanese word. That means savor in Japanese. Yes.
Sorry, I just want to make sure I didn't misunderstand this. When you're sick and you lose sense of smell, can't your taste buds change because one of your senses is blocked? No?
No, your taste doesn't change, but smell and taste combine together to create flavor. What I call flavor, that you call taste. Because you put something in your mouth and you assume that what you're perceiving is just from your taste in your mouth, but it's also triggering your smell. And smell actually plays probably a more important role in flavor than taste, which is weird, I know. But like with COVID, where people lose their taste, they're really losing their sense of smell, and that's impacting their taste. Their flavor. So I try and separate those out. Taste, they're strictly signals coming from the tongue. Flavor is our overall perception of what we're eating, which not only is smell and taste, but it's also vision, right? If I give you two things of ketchup to dip your french fries in, and one's black and one's red, are you going to have us put food coloring in the one's black? Taste exactly the same, but vision actually contributes to how we perceive flavor. Yes, Liz?
And that's why they tell you, like, when you have something gross, to like, hold your nose.
Yeah. Yeah. So you got to take this nasty medicine, you hold your nose, take it, it's going to be less flavorful. Right? Okay. So, receptors for smell. Receptors are found way up in your nasal cavity, right? Past a bunch of bones in here, and they're in what we call a mucous membrane up here. And it's interesting because of all the sensory receptors in our body, smell is the only one that are actually neurons, right? So we have rods and cones that are those kind of modified cells. We have these hair cells in the vestibular system, and in the hearing, we have 4 different mechanoreceptors in our skin, right? But here, these are actually neurons, and their dendrites are bigger, almost like finger-like projections, and they have receptors on them in this mucus line. All right. The mucus sort of protects them from drying out. They're not just hanging out there in the air. All right. Transduction for olfaction. Those receptors that are on those olfactory neurons are metabotropic receptors. They're just like the ones we talked about. Lock and key mechanism to release a G protein inside of the cell. Except here, it's not neurotransmitters activating them, it's smells, it's chemical shapes, it's the shape of chemicals in the environment. So we can have a smell that might activate multiple receptors. Different parts of that shape might activate different receptors. And we have 1,000 different shapes, receptor shapes that can be stimulated. Now, what's interesting is we can have chemicals here that are very similar, okay? The only difference in these two chemicals is that an oxygen has a double bond to this carbon here, and instead of a double bond to an oxygen, this one has 2 hydrogens sticking out here. Okay? This stimulates receptors in our nose and smells very musky, very animal-like musk. And this, it can't stimulate any receptors. We have no perception of it. Just that one little molecule change makes this one not able to bind to anything in our nose, and this one binds to a bunch of stuff to create that musky stuff. Here we have 2 that are very, very different in shape. They both smell like pineapple, and we can't discriminate between those 2 chemicals. So it's really complex with olfaction because we have 1,000 different places it can bind, and that shape can activate multiple receptors, right? So it becomes this pattern of receptor activation that we associate with a particular smell. Smell is also interesting because it's like, what are the primary tastes? Sour, sweet, bitter, salt, umami, right? What are the primary smells? Name some smells. Bad and good. Huh?
Rotten. Smell less messy.
Okay. So if I ask you, what's your favorite smell? What's your favorite smell? Cinnamon? What else? Just name some smells for me.
Lemon.
Huh?
Cookies in the oven.
Cookies. Your grandma's house.
Vanilla.
Vanilla.
Lavender.
Everything you're naming is the thing that produces that smell. Our perception of smells are memories, are things. Grass, roses, right? We don't have primary smells. We can't break them down into categories, right? We can say, describe the primary taste of lemonade. It's sweet, it's sour, right? You can break it down into components. Describe the primary components of a rose. It's just a rose. You just know it is because it's associated with that smell. Yes, Chris?
Is that kind of like when people lose their smell from COVID, like censoring, you like tap into the memory of what that's like how you remember that smelling to like research it?
Yeah, right? And that can produce what we call cognitive dissonance, right? Where now that's not matching up with the right memory. All right. So those receptors, I'll jump into the pathway just a little bit. Those receptors are sending their output to the olfactory bulb. That's why you had to learn that. That thing's sitting out there on the sheep brain. Okay? The receptors send their axons to the olfactory bulb, and they stimulate places in the olfactory bulb. And that pattern of activation represents that chemical. That chemical. Okay? So, and those things are called glomeruli. You don't really need to know that term. But different glomeruli are activated by each of those labeled line receptors. And it produces a pattern of activation that's different for butanol, pentanol, and propanol, and we can discriminate between those three different smells. Okay? Now, so this is the cross-fiber pattern. We know this pattern of stimulation represents pentanol, and this different pattern of stimulation represents this. Now we call this cut grass and a rose. Right? It's just two different things stimulated. Right? The thing is if I was about to make a really bad point. If you're out cutting the grass and you have that cut grass smell, and you stop to smell the roses, right? You smell a rose, and you have grass and rose, they don't interfere with each other. But this is what's happening in your olfactory bulb. They're both coming in at the same time and stimulating, yet somehow our brain teases that apart into different objects. Like, we don't know how our brain does that. It's pretty amazing. Right? How many of you ever been to Papa's? The breakfast place? You ever been to Papa's?
Papa's breakfast? Oh, Papa's.
Yeah, Papa's breakfast.
Don't eat there.
So when you walk in there, what do you smell? Bacon, coffee. Like back when I went to school here, smoke, because we smoked in there. You can't do that in restaurants anymore. Right? But all that stuff is assaulting your nose. You walk in that door and it assaults your nose all at once, yet they're discrete. All the chemicals are mixed up in the air together. It's somehow our brain can pull them apart into separate perceptions. Yes, sir?
It might not be related and you don't have to answer it, but like when you walk into a place like that and it's like a lot of smell all at once, but then you kind of adjust.
Yeah, that's called adaptation. Yep. It's like when you walk into your roommate's room and it smells really bad, you just sit there and tolerate it, it'll go away.
What
Yes.
you were just describing is a cross-fiber pattern.
Yes. Label line from the specific receptors that are being stimulated, but the brain's looking at the pattern to recognize the stimulus.
So you really can't eat nose line as the commercials call it?
Yeah.
Really? Okay.
Yeah.
Either line.
Okay, pathway. Olfactory bulb goes to the olfactory nerve, goes into the amygdala and thalamus. The part of the thalamus is the medial dorsal nucleus. The thalamus, right? What's the amygdala involved with? Emotion. Do smells generate emotion? Positive and negative mood? Yes, right? Almost all, it's hard to find neutral smells, right? Smells go straight into the amygdala and are stimulating emotional matter. Okay? From the thalamus, it goes to this area called the piriform cortex in the frontal lobe, and it also stimulates the limbic system and memory. Right? So smells, the primary perception of smells are memory and emotion. Right? And you can smell your significant other's cologne, and that generates a very positive emotion until you break up, and now you get a very negative emotion, right? So we can flip a switch on that value in the amygdala depending on experience. All right. This is just another example of the pathways. Comes into the olfactory bulb, from the olfactory bulb goes into these areas. I like, this is probably the easier one to focus on, slide 23. Okay? All right. The primary area is the piriform cortex, but there's a secondary area called the orbital frontal cortex, or OFC. That's where flavor is. That's where flavor is. OFC. Secondary area and function, OFC, flavor. Right? We also have association cortices like the amygdala for emotion, the hippocampus for memory. Okay. Smells, when we talk about memory, smells are directly tied into episodic memory, things that happened, right? But not really to declarative memory. So there's been a lot of studies like, hey, if I have peppermint in my study room while I'm studying, and then I chew peppermint gum, will that smell help me remember everything I learned? It doesn't. Don't waste your time. Declarative facts are not linked to smell. You might remember the room you were in, and what clothes you were wearing, and what time it was, and who was there. Like all the episodic memory can get linked to smell. It's difficult to link that to declarative.
What's the function of the
That's, we don't really know. Because normally it would be organized by column for features, but we don't really know because it's not that way. It's not. Maybe it's just a way to get it into the memory and emotion. All right. I gotta get through this. There is a small, in non-human mammals, there's a second olfactory system called the bonal nasal signal system that measures pheromone, that receives pheromones. Okay? You know what pheromones are? Chemicals given off by other animals, right? And they provide particularly reproductive signaling things. And so there's been a lot of debate, do humans detect pheromones? And we don't have a functioning bonal nasal organ. There's traces of what we think it might have been, but in your nasal cavity, but it's not hooked up to anything to go to your brain. So pheromones are chemicals given off by an animal that have another effect on another animal. Okay? Are there human pheromones? Well, some people want you to believe there are. The Axe commercials, you put Axe on, and all of a sudden it's like a magnet, right? And that's pheromone communication. And in animals, it works that way. In humans, not so much. Okay? So there are odorless chemicals that our brain can detect. We think that it does that, though, through our regular olfactory system, that can produce physiological changes. Right? But it doesn't produce the overt behavioral responses that we see with animals. So there's not an overt sexual response or behavioral response. When I say that, Ortho lab at Florida State when I was there, it wasn't my primary lab, I just rotated through it, but they were studying the nasal organ with hamsters, and they could take a vaginal swab from a hamster that was receptive for mating and stick that Q-tip in a cage with a male hamster, and the male hamster would copulate with the Q-tip. It's that over response. Doesn't even have to be an animal, right? It's a driven thing. So humans, no, we don't have stuff like that. It's not like Max commercial, but we do have some detection of it.
The book kind of talked about like how if a woman's smell is like split from a male, it can increase their fear, like stress hormones. I don't know if
Yeah, so there are chemicals that can cause changes, right? But not overt behavioral responses. Okay. So we do have a mechanism to detect that. And in fact, women that live together often start to sync up their reproductive cycles, and this happens through this thing, right? And they did a study, it's not a long time ago, women put pads under their armpits, and then other women in different dorms smelled those, and they got the two dorms to cycle up their reproductive systems. Don't have time for all this. Okay. So we don't have to have a functional vocal nasal organ. Our olfactory system, it looks like, can do some of this, but not like animals. All right, taste in 10 minutes or less. My favorite sensory system is getting jokes. All right, so taste buds in your tongue contain these onion-like collections of cells that have an apical side where taste interacts with transduction mechanisms, and a basement or basolateral side where they send action potentials up to the brain. That's all you need to know about that. Okay? Interesting thing about taste cells is that they turn over, they die and get replaced about every 10 to 14 days. So the taste buds you have in your mouth right now did not exist when you started taking this course. All right? They turn over every 10 to 14 days. Which is kind of amazing because our sense of taste stays very stationary. I mean, it doesn't change even though these cells are constantly turning over. We don't really know why that happens either. All right, transduction. Salt comes in an ion channel, just like an EPSP. When you put a salty pretzel in your mouth, that salt, that sodium comes in and depolarizes the cell through an ionotropic channel. Sours are primarily acids. They have hydrogen molecules. The hydrogen molecule blocks the potassium channel, keeps potassium from leaving, causes depolarization. So salt and sour both use ion channels to get transduced. Everything else, sweet, bitter, umami, all act through metabotropic locking key mechanisms. It's how we can create an artificial sweetener, a chemical that has no calories but binds to the sweet receptor that produces sweet taste. Just like sucrose would, but it doesn't have calories. No? No? Good. Okay. We think, particularly my lab early on, we demonstrated evidence that we can detect the chemicals that are found in dietary fats. And other labs have looked at the transduction mechanisms and said that fats are both ionotropic and metabotropic. I'm not going to get into that. It's not enough time. All right, neural coding. Here we see a nerve, a neuron, and we put salt, we put sweet, we put sour, we put bitter. This is water. What is this? How is this neuron responding? What is it responding to? Is it responding to salt? Not really, right? Is it responding to sweet? Yes. All those action potentials there, right? So that would be an example of a label by neuron. It really is only responding to one thing, right? It's a sweet. What about this neuron? Here's salt, here's sour, here's bitter, here's sweet. What is it responding to? Salt, sour, and bitter, right? Is the pattern different for each of those? If you just look at that, can you see a difference in the pattern? right? Kind of medium activation, heavy activation. This is kind of an on-off, on-off, on-off signal, right? So we have both the cross-fiber pattern signals and we have some labeled line. Labeled line is only for sweet and for salt. So here we see this neuron only fires the sweet, doesn't fire the salt, sour, or bitter. This one here only fires the salt, doesn't fire anything else. And then we have some cross-fiber pattern neurons that are going to respond broadly across sweet, salt, and bitter. We have to look at the pattern in order to see what those are. All right. Why do you think we have labeled line just for sweet and salt? Yes, because the two things we really need to consume to live is glucose, sweet things that can be converted easily into glucose, and salt. We can't make salt, and you know if your salt balance gets off, you can't send action potentials, right? So those are two things we want to really detect. Then the rest of this is kind of like, okay, is that a good or bad sour, bitter salt?
So that's why it's labeled line.
Yes, because we want to detect those things. That's an adaptation, so when you are adapted to something, and then you put something else in, it interferes with that. Yes?
Any of them can be cross-fiber pattern?
Sweet's really not. It's really discriminating between, sweet is just labeled line. It's sour, bitter, salt. All right. We actually have 3 nerves coming from our tongue. Cranial nerves 7, 9, and 10. They come into the brainstem, an area called the NST, nucleus of solitary tract. That then goes, has connections to both the hypothalamus and the amygdala. What does the hypothalamus do? Why do we send taste directly there? Hunger and satiety, right? And the hypothalamus, notice that's a bidirectional signal, right? If you're really hungry, things taste better. If you're really full, things don't taste good. The amygdala, again, foods have value, have emotional value. You can have cravings and aversions for food, positive and negative value. The VPM of the thalamus, and then it goes to the insular cortex, is the primary area, and there we have columns for sweet, sour, salt, bitter, and umami. And from there, then it goes to the orbital frontal cortex for flame.
What is
Yeah.
What is NST?
It's actually nucleus of the solitary tract. It's sometimes also written solitary nucleus.
So the primary area is the insular and frontal overkill.
Yeah, this insular cortex will be great.
Okay.
Insular.
And the secondary areas are the other ones you mentioned, the amygdala and hypothalamus.
Yes, and particularly the orbital frontal cortex for flame. All right. Hypothalamus, hunger and satiety. This area, PDN and amygdala, is for, the PDN is really cravings and aversions, we think, and the amygdala is emotional value. Last thing I want to talk about, sort of complex behavior, conditioned taste aversions, right? Go out and you eat a bunch of cheeseburgers, really yummy, good cheeseburger. Go to Heights, it's good, right? But then you get the flu, not the coronavirus, hopefully it's the flu, and you get really sick, right? And now, how do you feel the next day about those cheeseburgers? Did the cheeseburger make you sick? You know you got the flu. You know you didn't get food poisoning, right? You know anything to, does anyone have a taste aversion when they were a kid? Anyone want to share what it is? Yeah.
We had crispy cream, and I was carrying it in the car, and I lived like 10 hours away. I don't know why I got caught with us, but I got car sick, so now I
You feel sick, yeah? Good.
My whole family got sick from pot herbs and I don't understand it anymore. I was like 4 or something.
Yeah.
When I was a kid, I had like ADHD meds, and my mom had it in like peanut butter and stuff, so like I could barely stand peanut butter for years. Just like, no.
The Charlie octopus things.
I don't know what that is, but okay.
They're like, you know, like Charlie sourworms?
Okay.
It's in chicken octopus.
Okay. So they can be very specific, right? It's that thing that you associate with being sick, right? So conditioned taste aversions can be a problem when we have situations where people are repeatedly getting sick, right? Like chemotherapy. And so we have to work really hard and try and not associate foods with things that are getting us sick. So lesion studies have shown that this PDN and the amygdala are the parts of the brain doing this conditioned taste aversion. Why does the cheeseburger physically not taste good now? Because a switch got flipped in the PDN to say, hey, that incoming taste signal used to be good, now not good. All right, we're good? You made it. Workshop tomorrow. Bring your lab report, either print it out or on your laptop where you can work on it. I'm going to have a worksheet for you to work through. I'm going to be there to answer questions. TA's going to be there to answer questions. The goal of tomorrow is that you leave with your lab report either done or you know the minor revisions you have to do in order to have it done so you